Alright, ladies and gentlemen, this is going to be our next video in biology, and specifically going into the details associated with DNA replication. Let's get started on this next video. So why does DNA go through and replicate here? Well, DNA is going to need to go through and replicate in order to go through mitosis. And really what we're going through and looking at is the details which are occurring in the synthesis phase of the cell cycle. So we see replication going through and occurring in the synthesis phase, and DNA is going to replicate whether or not we go through mitosis or meiosis. Now, if we're thinking about the process of mitosis, we need to make two cells with exact copies of those DNA. So when those two cells go through and arise, we have the duplicate set of DNA, which is important for when we go through and create other cells or multiply those cells in our body. Same thing for meiosis, except we're making gametes. So when we talk about what replication is, well, DNA replication is the process in which DNA copies itself. It's really that simple. So we're going to go from one set of DNA into two sets of DNA for our two separate cells. And the basic and the gist of this process is going to be, we're going to take parent DNA, we're going to go through and separate that DNA, and then we're going to use each of the sides as templates to go through and make our two daughter DNA. We went from one, split it in half, add the templates in there, and add our new strands. So now we have two sets of daughter DNA. And this daughter DNA is going to be made out of one original strand and one new strand. So we're going to introduce a concept here called semi-conservative. This is where half of the parent DNA of the molecule is conserved in each of the two daughter DNA molecules. We have our original strand and our new strand, so a semi amount or part of that DNA is original, conserved, and part of it is brand new. Now replication is going to go through and occur. Well, where is it going to occur? It's going to occur in the nucleus of eukaryotic cells, and in prokaryotic cells it's going to go through and occur in the cytoplasm. Remember, prokaryotic cells don't have a nucleus, so we're just going to see it occur there. And this is an important concept because remember, that is where the DNA is, and that's going to be its general location. So where is replication going through and occurring? In eukaryotic cells, it's in the nucleus. In prokaryotic cells, it's in the cytoplasm. Now, before we go through and introduce the process of replication, we're going to need to understand the three players. There are going to be three players and three enzymes. We're going to have helicase, which is going to be enzyme number 1. It is going to unwind the DNA. We are then going to have primase, which is going to go through and lay down the primers. And then lastly here, we have DNA polymerase, and that is going to add the daughter strands of DNA and use the original strand as a template to go through and make our new set of DNA here. So before we understand anything, we need to understand this is an enzyme catalyzed reaction, and it's going through and occurring via these three main enzymes. All right, so let's go through and delve into the details associated with replication. So replication is going to start with step number 1 here. Helicase is going to unwind the DNA double helix, break the hydrogen bonds between each of the nucleotides, so here is our helicase enzyme, and go through and create an opening in those two strands. So what we can see here is helicase is going to move through, and it's going to create what we call a replication fork here. What this is doing is separating out the DNA into two pieces, almost like a fork, so we can then use the original strand here as a template now. Now step 2 is going to be, we're going to need to add primers to this DNA. So primers are going to bind to the leading strand. This is going to be the leading strand along the top, and DNA polymerase is going to bind to the primer site. The lagging strand's on the bottom, and we'll talk about why the lagging strand is different than the leading strand. But for right now, we're going to see this enzyme here, primase, is going to go through and add what we call a primer. A primer is basically just like a little flag that indicates to another enzyme here, which we're going to see in a second, DNA polymerase, a starting site for that replication. So the goal of the primer is just to create a flag and create a starting site for replication to occur and DNA polymerase to know where to go. So primase is going to go through, lay down that primer, and then our next step here is DNA polymerase is going to copy the daughter strand of the leading strand. So it knows where to go through and bind because of that primer. And it's going to follow the template, right? We have that yellow template there. We know A goes to T and G goes to C. So we know what to lay from the free nucleotides in the cytoplasm, and we can make that leading strand. So helicase is going to continue to unwind, and then we're going to see DNA polymerase go through and follow behind helicase and follow along that leading strand. Now the leading strand is the easier strand to go through and create because it only requires one primer segment. The lagging strand is a little bit different. The lagging strand is going to move, if we're looking from right to left, from 5' to 3', whereas the leading strand is going to be from 3' to 5'. Because the leading strand is 3' to 5', the DNA polymerase can go through and follow right behind the helicase and go through and add those segments there. The lagging strand is going to be a little bit different. Primase is going to have to go through and lay primers, in this case 5' moving down towards 3' here. And when it goes through and does that, DNA polymerase is going to have to go through and fill in these gaps here called Okazaki fragments. These Okazaki fragments are going to go through, you see our primer here where our DNA polymerase adds, are going to be what is going to have to be filled in. DNA polymerase can only move 3' to 5'. And in this case here, because our lagging strand is 5' to 3', if we're moving right to left, and we want DNA polymerase to move to the right, it has to go through and add in those segments in this fashion. So basically the difference is the leading strand is going to follow along and do it in one continuous strand along the replication fork, whereas the lagging strand is going to be in these Okazaki fragments. So again, if we look at this in a little bit more detail, we can see 3' to 5'. This is how DNA polymerase is going to go through and move. So primase is going to go through and add the primers in here. It's going to add a primer here, a primer here. DNA polymerase is then going to fill in these gaps between these Okazaki fragments, and as it fills in these gaps between these Okazaki fragments, it's using the primer as a flag to go through and know where to go through and add the new strand of that DNA. So if we look at the parts of the DNA here, we can see here's our parent strand, here is our daughter strand, and this is again why we call it semi-conservative. We're using the parent strand to go through and create a template for the daughter strand. If we look at these other sections of DNA along the daughter strand, we have our primers there. And our primers are going through and allowing us to have little sections and flags so DNA polymerase knows where to move. So if we look at this process in just its basic form, this is what's going through and happening. We're using the semi-conservative principle here, where we go through and we're going to unwind the parent DNA. We're going to have two original strands that are going to go through and be separated that are going to be used as template strands to create two of the new molecules that we see here. This process, this semi-conservative process, is what we see for DNA replication. All right, so there's a lot of information in that video, and this is why we go through and there is a DNA review video, but we need to understand a couple of things here. We need to understand what replication is. We need to understand why cells are going to go through and replicate, what semi-conservative and anti-parallel mean, and do we understand the overall details and the process of replication. This is going to be the end of the video. We'll see you all in class tomorrow.